So good evening. Thank you everyone for joining us. This is an AONM webinar on TOXIPLEX, and it's a direct mycotoxin detection assay for human serum and plasma. And we're delighted to welcome the two developers of this, together with their team, Professor Leona Gilbert and Mr. Kunal Garg. I'll just introduce Mr. Garg because he's giving the presentation this evening, primarily, though Professor Gilbert will be available to answer questions at the end. And we're also very pleased to have Marcus Berger from AONM'S here with us, who will also be helping with the questions at the end. So just to tell you a little bit about Mr. Garg. He's the co-founder and chief technology officer at Tezted in Finland, which is the company that develops these diagnostic tools. He graduated from VIT University, India in 2013 and acquired a master's in neurosciences from Jyväskylä University in Finland in 2015. He has an academic background in biotechnology, and his experience as an entrepreneur has provided him with a broad range of skill sets to create and commercialize in vitro diagnostics. You'll probably be familiar with TOXIPLEX, which AONM has been running together with AONM'S for many years. And that was a development of Professor Gilbert's and Kunal's, a diagnostic tool to simultaneously test several pathogens and multiple disease stages in patients suspected of tick-borne disease. And this that he'll be introducing today, TOXIPLEX, is a tool to help directly detect and confirm fungal toxins in human beings. So we'll be hearing all about that for about 40 minutes from Kunal. And after that, answer any questions you might have that you're welcome to put into either the chat or the Q&A. So over to you, Kunal, and thank you very much indeed, everyone, for being here, and Kunal for tonight.
Great. Thank you for the introduction, Gillian. I was informed that we have almost 100 attendees today. Thank you all for participating in the webinar. We do have a very interesting topic today, which is the detection of mycotoxins in human serum or plasma, especially in the context of our new product, which is TOXIPLEX BASIC. And for this presentation, I wanted to broach the topic of detection by understanding the different modes of exposure to mycotoxins and their impact on our health. Also understand the advantages and disadvantages of the existing methods. And finally, the reason why we are here is to also understand what we've done to design TOXIPLEX BASIC differently from the existing tests. So to begin with, when we talk about the modes of exposure to mycotoxins, it is quite well established that when we eat any food that is contaminated with a certain species of fungi, we are also ingesting mycotoxins. And here is a quote from the World Health Organization recognizing the same. So when you would go to the websites of US FDA or the Food Standards Agency in the UK, you will always see certain common mycotoxins that are always of concern. And they include aflatoxin, deoxynivalenol, fumonisin, patulin, and ochratoxin, and then also a few like zearalenone, and the list goes on. But there is also emerging evidence to indicate that we can get exposed to these mycotoxins through inhalation in indoor environment, for example, in water-damaged buildings. And this is a research published recently in 2022, wherein researchers in Germany sampled mold-infested building material from households in northern Germany. And this material included wallpapers, isolation material, glass wool, etc. And they found 5 important species or significant species of fungi, which included Penicillium, the family of Aspergillus, Acremonium, Stachybotrys, and Fusarium. And if you look at the mycotoxins that these fungal species produce, many of them actually overlap with the mycotoxins of concern that I showed in my previous slide, which we can get exposed to by eating contaminated food. For example, penicillium produces patulin or ochratoxin A. The family of Aspergillus fungi can produce aflatoxin, ochratoxin, gliotoxin, and sterigmatocystin, which is actually one of the most important mycotoxins that has been coming up in the context of indoor environments. The sterigmatocystin mycotoxin is also produced by Acremonium and Stachybotrys species of fungi, and also a few other mycotoxins such as satratoxin and roridin. Finally, we have the Fusarium species, which can produce different forms of fumonisin or inianitins. The name of these mycotoxins are quite difficult, but these are the mycotoxins. So when we talk about the indoor environment, we are obviously not focused on just households, because people can get exposed to these mycotoxins at their place of work. And again, there's a recent article which from Tehran in Iran, wherein the researchers went to a wet waste management company, and they measured the air, the surfaces, and human serum for aflatoxin B1. So they essentially collected each vial, and they measured through UHPLC. And what they found at the end was that the levels of aflatoxin B1 albumin was significantly higher in spring and in fall in people that were working at the wet waste management company versus the people that were not working at the wet waste management. And this goes on. Now, the list that you see on your screens is not exhaustive by any means. As I said, the evidence is still emerging, but there is evidence to indicate that in building materials such as wallpaper, cardboard, you can find aflatoxin, DAWN, or OTA, and more in the industrial sense, you can find zearalenone. There are blog posts to indicate that it can also be associated with water damaged buildings. But I found a peer-reviewed article indicating that in the industrial settings, it can be something that you see in the corn dust elevators. When it comes to fumonisin has been shown to be eluting in the urine of humans and animals in various indoor settings. This also includes the industrial settings. Now, having a good understanding of the mycotoxins that are that we can get exposed to through food, through contaminated food, and through indoor environment, we wanted to focus on these 5 mycotoxins that you see here, and it is good to understand their impact on our health. So when it comes to aflatoxin B1, it can target our liver and can result in hepatocellular carcinoma. DAWN is a disruptor of gut microbiota and also our innate immunity. Fumonisin, on the other hand, it disrupts our adaptive immune response by inhibiting the maturation of dendritic cells, and it also results in cytokine secretion. When it comes to ochratoxin has been recorded to cause nephrotoxicity and also secrete pro-inflammatory cytokines. And finally, ZEA, it can inhibit the synthesis of very important blocks such as proteins and DNA. Having understood the mycotoxins of concern, the way we get exposed to these mycotoxins and their impact on our health, I think it would be great to look at the methods we can use for detection of mycotoxins. So when it comes to the detection of mycotoxins, I think it is a good idea to understand the whole spectrum of detection of fungi or fungal infections in humans, and also the detection of mycotoxins. So when it comes to culture-based methods are the gold standard for detecting fungal infections, but 50% of the time they miss documented cases, and it takes a lot of time to get the results. And fungal and culture-based methods are not the technique for mycotoxin detection. The same applies for molecular-based methods. Unlike culture-based methods, molecular-based methods do not need a live fungal cell for the detection of fungal infection, but they do result in wide lab-to-lab variation in results. Again, molecular-based techniques are not something that you can use for mycotoxin detection. However, what you can use for mycotoxin detection and has been used quite widely are the chromatography-based methods. These are gold standard for mycotoxin detection because you can detect multiple mycotoxins with high accuracy and reproducibility. But the other issue with chromatography-based methods is that the machinery is quite expensive. It is not available for everyone. And this is one of the reasons why mycotoxin testing in humans has not really taken off. On the other end, when you do have such machinery, you also need really high level of expertise to operate such machinery, to get the results out and to interpret them correctly. Antibody-based methods, and when I say antibody-based methods, I'm referring to the direct detection of the mycotoxins rather than measuring the antibody responses such as IgG or IgE. Antibody-based methods have been very well established, at least in the food sector, wherein they help detect one mycotoxin at a time. They don't need high level of expertise and they are accessible for everyone. But like any other antibody test, antibody-based methods are prone to cross-reaction. And finally, we have the aptamer-based methods. The aptamer-based methods do offer the same advantages as the antibody-based methods. However, they are more robust. But the bigger disadvantage with aptamer-based methods is that they are also prone to cross-reaction. They're not commercially available at the moment. And the method that is used for aptamer selection, which is the SELEX method, it is in itself a very expensive endeavor. So what are we really doing different with TOXIPLEX for the detection of mycotoxins in humans? So I've been told from our great partners at AONM and at AONM'S Labs that I need to make this very, very clear, is that with TOXIPLEX BASIC, although it is an immunochemical assay or immunoassay, we are not detecting IgG, IgE, or IgA response. We are detecting the molecule directly. And this matters because there's always a question of if the patient is suffering from a different condition and how that deals or how that brings into question the immune status of this patient. If the patient is immune compromised, for example, can the patient actually mount an antibody response? So we are not taking that into account. We are really taking into account, is aflatoxin present in the human serum? Is DAWN present in the human serum? And so that is what we are doing. We are not detecting the IgM, IgG, or any form of antibody response against mycotoxins. In fact, we are detecting the mycotoxins themselves. And the second thing that we're doing unique is that we are not testing for mycotoxins in urine. We are testing for mycotoxins in human serum or plasma. And a few reasons for this is because if you look at the literature, the use of human serum or plasma for measuring mycotoxins in humans is 5 times more common. There is a good reason for this, because when you use urine, you have to do a creatinine normalization, because the urine volume from one individual to the other is not going to be the same. So you need some form of measurement to standardize the outcome. And the other reason is that the mycotoxin intake on a daily basis is different. So once you've done the mycotoxin testing, you need to sample another test in within 24 hours. So we are not looking, so just clarify, we are looking for mycotoxins directly in human serum. So the principle for the assay is essentially that we bind antibodies that are very mycotoxin specific to an ELISA well. When our manufactured test goes to Armin Labs, for example, they essentially extract the mycotoxin from the patient sample and they put it on the plate. Now, over here, you see the mycotoxin in black, and then the mycotoxin with a conjugate. The mycotoxin in the black and the mycotoxin with the conjugate are going to compete for the binding space on these antibodies that are bound to the plate. Once we've washed the unbound mycotoxins or the mycotoxin conjugates, at the end, we are left with whatever had the higher concentration. If the mycotoxin extracted from the human serum was high in concentration, you would see more of them binding to the antibodies. So what this exactly means is that if the mycotoxin from the human serum sample is in higher concentration, the mycotoxin conjugated to, for example, horseradish peroxidase is not going to bind, and there is not going to be a signal, which essentially means that patients having low signal actually demonstrate high levels of mycotoxins, and patients with high levels of signal actually demonstrate low or no levels of mycotoxins. And we are the first of its kind ELISA, because most of the ELISAs in the market at the moment are not focused on these 5 common mycotoxins. And we have shown through our research and development that our accuracy is about 93%, and our lower limit of detection goes all the way down to 0.15. I will be showing more results regarding this. But here is our paper. If you're interested in reading it, is an open access paper, and you can see all our validation results in this paper. But I think it is worthwhile getting into some of the key highlights from our paper, which, for example, is the low limit of detection, which essentially means how low can we really detect a mycotoxin. In the red over here, you can see the lower limit of detection for each of the mycotoxins on TOXIPLEX BASIC. We have compared our test results with food, with tests designed to measure mycotoxins in food. And the reason for this is because we did not find in the literature the form of ELISA that we've designed to compare against. But we decided to compare our test against food-related tests, because the principle of ELISA remains the same, and the food-related tests are well established. So you can see that for food-related aflatoxin, it can go anywhere. The commercial can go anywhere from 2 to 1.5, and we are presenting a 0.61 limit of detection. For DAWN, it can go anywhere from 100 to 250, and we are presenting 19.53, which is quite low. FOAM, for example, is presenting 4.88 limit of detection, and in commercial tests, you see between 30 to 200, for example. And OTA is definitely an area that we are working, but we see 19.53 as the limit of detection. I have to say, while developing this test, I was surprised to see 0.15 as the lower limit of detection for Zoya, because I've not seen that in emerging technology for food or even in the commercial tests. And of course, we've repeated our tests on multiple occasions to make sure that our results are representative on a daily basis. If you remember in the previous slide, I mentioned that antibody tests are prone to cross-reaction. And what we've done over here is we've interacted different mycotoxins in our test against different antibodies on our test. And as you would expect, a aflatoxin antibody to react with aflatoxin, and that is why you see 100% reaction. But in the terms of aflatoxin antibody, there's only about 2% cross-reaction with other mycotoxins. And this is within the range that we see for antibody tests, even in the food sector, wherein the antibody tests are well established. For DAWN, it is between the cross-reaction with other mycotoxins is between 8 to 11%. For fumonisin antibody, it is 4%. For ochratoxin antibody, it is between 8 to 11% again. And for ZEA, it is between 3 to 4%. And these levels are within what we would expect to see in an antibody test. So there's not much to be surprised by over here. So when you do order TOXIPLEX BASIC, hopefully, this is kind of the example report that you get from AONM'S labs, wherein you have the patient information, and then you have if the mycotoxin has been detected or not. We indicate the lower limit of detection in areas when the mycotoxin has not been detected. For example, aflatoxin B1 in this example report has not been detected because the level detected is less than 0.61. If you remember in the previous slide, I demonstrated that the limit of detection is 0.61. However, in the case of DAWN, the DAWN was detected because it was more than 19.53, and we quantify what the calculated concentration for the patient. The mycotoxins that are detected, then you get a brief report on what it actually means, and so this is kind of the example report. I have to say that we also report plate controls. We consider this as a very important part of designing our test kit, and AONM'S labs, for example, considers this very important as a daily routine, wherein we make sure that the positive control is positive and the negative control is negative, which essentially means that the plate is working as it should and the operator has performed the test as he or she should have. So in summary, mycotoxins, we can get exposed to these very, very small molecules through food and through indoor environments like water damaged buildings. There have been a lot of testing through gold standard, which is LC-MS. But as I discussed, these equipments are not accessible for everyone. On the other hand, we have rapid screening with antibody tests, but antibody tests up until now have been limited to a single mycotoxin. And we believe that we are bringing the best of both techniques with TOXIPLEX, wherein you can detect multiple mycotoxins with better and higher accuracy, and it is easily accessible for labs and for the patients and for the healthcare providers. So in the event that you're interested to test and order TOXIPLEX, you can always reach out to AONM or AONM'S labs, and I'm sure they will also follow up with information regarding the test and its features. And I can take questions at the moment, me and Leona. Thank you.
Thank you very much indeed, Kunal. Absolutely fascinating. Just before we go on to the questions, Professor Gilbert, I just wondered whether you could let us know what led you to take this very interesting development after doing the TICPLEX. What was it that actually sort of prompted you to take this path?
Well, full disclosure, this has been an incredible collaboration with Sanofi in Mexico, and we listened to their needs and their wants, and they were unsatisfied with the testing that they were using, and so we came up with solutions, and this was one of the ideas that we had. Of course, we also had the great kind of idea to use, UHPLC, so high performance liquid chromatography as well, but that didn't work out. So, as science and, test developments, goes, some things work and some things doesn't. And so this was a great collaboration with Sanoviv and Fausto and Flora there, great scientists, and we decided to, okay, let's kind of improve kind of the whole concept of testing for mycotoxins, and not just testing one microbe, one mycotoxin at a time, but also multiplex it, because that's kind of like our signature at Tezted. We want to multiplex detections or diagnostics so that we, we get a wider sense of what's going on in the patients, and this is why we multiplex this system. We have tried different aptamers, we've tried other antibodies and techniques, but of course, the bottleneck with this technique is the sensitivity of the antibodies that we use, obviously, and of course the reproducibility of people performing this test, and that's why we include those controls there to make sure that the plate itself is being performed accurately and reproducibly, like we have with TOXIPLEX as well. And AONM'S lab is showing that they are, performing the test kit quite robustly, and this is great for us. And we could also see it with our other partners as well, because it's a signature of our test that we have these controls there. Going from academic background into the industry, of course, we needed to make these tests, of course, routine lab friendly or, clinical lab friendly. But we still wanted to make sure that the controls were there. So if there's any doubt of the performance, well, we can look at those controls. So yes, so collaboration, and we're willing to collaborate with quite many people. Like we have a long history with the AONM'S lab collaborating and publishing our research and validating different kind of hypotheses that we may have. So yeah, that's how we did this.
No, thank you very, very much. Thank you. Just to correct earlier, I've been reminded, Kunal, that I might have said your master's was in neuroscience, and of course it's in the nanosciences. So I'm sorry if I got that wrong. It wasn't intentional.
No problem.
The question here is, one of them is, how many types of mycotoxin does this test detect? Can we say that it is those 5, or is it actually the sort of precursors and the downstream ones that are included as well, Kunal?
So it is those 5, but for example, aflatoxin is a precursor to steric metrocystin. So there is some form of relation over there. But then also we have to understand that, for example, aflatoxin B1, aflatoxin B2, when we look at the structure of these mycotoxins, the chemical structures, they're not very different. So if an individual is exposed to a different form of aflatoxin apart from B1, there is a possibility that we might also end up catching that on the test.
Okay, thank you. And another question here is, what are the clinical implications of a positive result on any of the mycotoxins?
And I would hand this over to AOMN because we cannot comment on clinical aspects, but what we can say is that if there's presence of mycotoxins, and depending on the amount, okay, and that's why it's really important that we demonstrate the amount of mycotoxin obviously will have stronger effect or lesser effect on the health. And so I would have to
Well, are you suggesting that it'll be actually quite useful after a period of therapy? And of course there are different approaches to this, and I mean, we will be having an answer to that question. We will definitely have therapists who've specialized in this for many, many years around, in order to give some ideas on how to approach these particular toxins. That's a wonderful, outlook. But are you suggesting that after 3 months or after 6 months, it would be worth testing and one can actually see the difference in level?
Yeah, I mean, and that's the whole point of recording these amounts of mycotoxin that are present, because therapy should, get rid of them. And of course, we all have kind of basal levels, and of course, this test kit, takes that into consideration. Of course,
yeah.
basal levels of mycotoxins that we're all exposed to. But if there's peaks in individuals of patients, and if they have a lot of mycotoxins in there, obviously that's going to have an adverse effect. Kunal gave a lot of citations to indicate that. But depending on the environment the patient is in, will indicate, of course, longer exposure and of course, the therapies and the success of therapies, of course.
Yes, thank you. And I believe Dr. Neil Nathan in the States, who talked at the Forum for Integrative Medicine a few weeks ago, said that he often notices one toxin as you kind of overcome that, another one might pop up a little bit higher after that. So that's something that it'll be interesting to note over time. I think we'll probably be taking very, very careful sort of monitoring, won't we, of the results and note whether that occurs in our patients. Marcus, that's something I'm sure that Armin Labs will be helping with. One doctor here who may not be from the UK, I do recognize the name, is asking, how do we get a test kit? Well, for the UK and Ireland, please do contact AONM. That was the last slide in the presentation. That's the helpline that you may well know, 0333 121 0305, which is on the website AONM.org. That's the Academy of Nutritional Medicine. And if you would like a test kit for Germany, then do by all means contact Armin Labs. That's arminlabs.com. And if you would like a test kit for the USA, which is, I believe, where this doctor is from, then maybe, Marcus, you could just let us know how that would work, the USA or Canada.
So Canada is already working to request the test kits directly through us, and US, as we are currently undergoing CAP and CLIA accreditation. So basically CAP accreditation is already achieved, and CLIA certification will be achieved within the next, we think like 1 or 2 weeks, as I just received another message by CLIA that our application is actually approved. So I think we're back maybe 1 or 2 weeks, and then you can also order our test kits again from US and also send it back to our laboratory. And what I wanted to mention earlier about the clinical implication, as you know, Armin Labs is familiar for or pretty well known for all of their checklists. We are probably also working on one for mycotoxins, so you get to know if there is a need to go for a mycotoxin test, and which one is likely to be there. So,
oh, that's brilliant. And then there is also a question about the cost of the test. We would recommend that you please have a look at the mycotoxin testing link on the Armin Labs tab on aonm.org, because there we will be putting the order form in the next couple of days. And in the meantime, if you'd like to call the number that I've just mentioned, then the price for the UK will be available, and the price from Germany from Armin Labs, and the price for the USA, I presume, Marcus, will be available very soon too. So it is a reasonably priced test, most definitely. The turnaround time is also being requested here. Marcus, I believe, and Leona and Kunal, you've also told us it's a very fast turnaround, isn't it? So we should be getting results within about a week, I believe. Would you back that up, Marcus?
7 to 10 days, including transportation time. So usually turnaround time for Armin Labs. Yeah.
Okay, great. Thank you. And could the test be affected by medications such as hydrocortisone?
So if I could answer this, and if other people want to put into, so hydrocortisone steroid actually affects the immune system. We're not detecting anything in regards to the immune system. I would expect that toxins, the flushing system with the kidneys and so forth, to flush out toxins. But if you're at the high levels that we can detect it, okay, or at the low levels that we can detect it, obviously there's the exposure level is greater. So Steroids will not affect this. We don't think the literature represents that it shouldn't affect this. And because we're really not looking at anything to do with the immune system, and steroids are there to help with the immune system. So.
Okay, thank you. How long do the mycotoxins remain in the blood and plasma after exposure to determine the accuracy of the test?
Yeah.
So I think the first thing I want to say over here is that we are not, we don't measure the accuracy of the test by measuring the mycotoxins in the serum or the blood. Since this is an analytical test, we actually spike a known amount of mycotoxin in human serum or plasma, and then exactly try to measure how much was spiked. So that is how we measure the accuracy of the test.
Yeah, but what is the life cycle of the mycotoxins that's circulating in the blood? So their mycotoxins are quite stable depending on your flushing system, and of course it goes back to first of all, plasma is more stable than urine, and traditionally with urine you need to freeze, and the freeze thaw of any kind of compound disrupts the bonds, the chemical bonds that hold the mycotoxins structure in place. And since we're using antibodies directed against the mycotoxins to detect them from plasma, of course, the confirmation is very important. So my point is that if you're constantly being exposed through buildings, through food, to these mycotoxins, and if we can detect it on a Monday, we're going to detect similar results on Tuesday, Wednesday, Thursday, Friday, if you take a blood draw each of those days. So they're quite stable because you're not getting rid of them. So we're detecting them. So you need to do therapies to get rid or change environment, not change environment, but obviously work on the environment or the exposure route that has given you these mycotoxins. So,
right, thank you. You've just mentioned all exposure to your environment. A question here is, does the test only identify mycotoxins in food, or can it also identify mycotoxins found in the air and the environment as well?
So yeah, go ahead, Kunal, sorry.
Yeah. So no, so we are detecting so does the mycotoxin in food, or does it identify mycotoxins in air and environment? No, we're detecting mycotoxins in human serum or plasma. So the matrix of interest is human serum or plasma. We are not detecting mycotoxins in air or in food. Those are different matrices. Those require different extraction process. Those require different optimized protocols, so to say. The test is currently optimized to detect mycotoxins in human serum or plasma.
Okay, thank you. There was a question, a very specific one here. Does this test detect mycophenolic acid, a mycotoxin from penicillium?
No, not at the moment.
Not directly, no. I mean, you had penicillium on your chart towards the beginning.
Yeah, we have certain precursors like the ochratoxin that are also produced by penicillium that we are detecting for, but not specifically for mycophenolic acid at the moment.
Okay. How does it compare to the Great Plains mycotox test? Is a question here. Sorry, I haven't finished reading it through, so I'll go to the end. Or has this been covered with the explanation regarding urine/serum differences? Great Plains testing is down at the moment, so we might be looking for a new one. So, I mean, I'm sure we don't really want to directly talk about competitive testing, but if you could just emphasize again the differences between yours and urine testing, Kunal, which you did go into, but maybe a little more detail.
Yeah, I think I can be scientific about it. The Great Plains has used LCMS, the liquid chromatography, which is the understanding that I have, and we are using the immunoassay. In the case of Great Plains, they are detecting many mycotoxins, but to the best of my knowledge, they are not detecting all the 5 common mycotoxins that we are detecting in TOXIPLEX BASIC. And then the other part is that we do not know the lower limit of detection for many of the mycotoxins detected by Great Plains, but in a few cases, like the ochratoxin and I think zearalenone, we are doing quite well in lower limit of detection. So scientifically and technically, although Great Plains is using a very nice machine, I think you can get quick results and for the 5 common mycotoxins through TOXIPLEX BASIC.
Okay, thank you. And this may partially overlap. Sorry, this one came in previously. The doctors ran a blood test to test for aspergillus. My understanding is that this test only rules out that this particular patient doesn't have an actual infection of mold in her body. This is someone who's unfortunately hospitalized at the moment and doesn't give any information regarding whether her health symptoms are the impact of long-term exposure to mold. Is the urine test the only way we can look for the presence of those toxins in her system, or should they have shown up in the bloods that have already been done? So that needs carefully sort of disentangling, but
Yeah,
it's somebody who hadn't yet seen your presentation. I hope they've been on it today.
I have to, ask a question to a question, but if the blood was done, I think it is important to see if the blood analysis was done to measure the antibodies, or was it done to measure the toxins in the blood? Because if the antibodies were measured, it is a very different test technique than what we are offering. So I think it is important to look into what kind of technique was used for the blood.
Sure, thank you. Well, I'll pass that back to the
And
mother. They may not have been able to watch. Leona, go ahead, please.
Sorry, I just wanted to also emphasize, we are not detecting mold in people or in the blood. We are detecting the mycotoxins, the toxins that come from those molds, from being exposed to it through eating it, breathing it, food feed, those kind of things. So it's really like we're detecting directly the mycotoxin within the blood.
Okay, thank you. Another question here. Do the toxins have an impact on IgA as well? I know you don't measure antibodies, but this is simply a question that's come in. Do you have anything to say about that? Is IgA, if it is measured, relevant? Or is that something you'd prefer to stay away from, because after all, we're not testing antibodies?
Yeah, we're not testing antibodies, but I would think that depending on where, again, we eat, we breathe, mycotoxins. So the first exposure, of course, is the mucosal area. And of course, antibody, especially IgA, takes some time to be made, but it doesn't mean that it is a clinical, that IgA has a clinical relevance. And we need to see long-term studies of correlating mycotoxins amounts in plasma or sera with these antibodies, because there is no correlation study. And this is, I think, one of the things that is exciting about this field. It's emerging, and AONM'S lab is getting their data sets, and also with AONM, we're going to get the data into formats that we can mine it, and so that we can maybe possibly see these correlations of that. And I think with more research, we can maybe, provide a better sound answer to this. But again, antibodies are not a signature clinical presentation at the moment, because it's mucosal and it takes time to develop that IgA. Yeah.
Right. There is a question here also regarding whether it might be available as a finger prick test at some point, because some patients find it hard to have blood drawn.
Oh, yeah. So Kunal, if you want to.
Yeah, it is in development at the moment. We have discussed some plans with AONM'S labs. We are figuring out some important logistics around recruiting samples, but it is under development, and we do hope to get it out.
Yeah, and we have to understand that the blood and urine, it doesn't matter if you go to any type of mycotoxin direct testing, you still have to extract the mycotoxin from your matrix, so from your urine, or extract it from your plasma and sera. So it's kind of like that dilution effect, right? We need a proper amount of blood, plasma, sera, okay, to do the extraction. Same, you need the proper amount for the urines, okay? That's really representative of the matrix of what we could find, no matter if we take, 1 ml from, the top of the sample, 1 ml from the bottom of the sample, we need a good representation. And that's the thing that we have to remember. There's an extraction protocol that we need to do before we actually do the test. And the actual amount on the test, I believe, of the extracted mycotoxins are 50 microliters. Is it? Yeah. So it's quite small, but it came from 500 microliters of serum. And to get 500 microliters of serum is quite simple for us, of course, compared to, the milliliters that you need for urine testing. But so the extraction is quite important for us. So if we're doing a finger prick or a blood spot, we have to make sure that we have the sensitivity and reproducibility and robustness of what we have right now compared to the finger prick or the blood spot.
Absolutely, yes. Yeah. Another question also, when you're getting toxins, mycotoxins from food, but you're perhaps not eating that food constantly, and then you move on to a different type of diet, perhaps it's just 1 or 2 evenings in a restaurant, will that disappear from the blood and not be visible the next week? Or is it because it's mycotoxins rather than the mold itself? Will that remain for a lot longer? Do we know that?
Well, again, mycotoxins have a life cycle, and I mean, we can do a quick Google search. I can't remember what it is at the moment, so please.
It's different for each probably mycotoxin as well, isn't it? Yeah,
it is. And if you have a proper kidney function and gastro function, you're going to clear it. You should, of course, and that's why we all have basal levels. We're all exposed to mycotoxins at some kind of basal level, anyways. And the point is that we need to go above that basal level, okay, because we're not experiencing clinical symptoms because of our basal levels of mycotoxins. So the point is that if the test is built and validated on above the basal level, so that we can really see that if it is positive, then it's, there is a clinical relevance to it. And I think that's what we have to keep in mind for ourselves, that traditionally basal levels are not harming us. Most of us aren't experiencing those symptoms coming from mycotoxins because we're functioning well. But of course, in really, with patients that have long-term consequences of many kind of diseases and ailments, of course, their kidney functions and other functions may not, you resolve the clearance of the mycotoxins, so they will stick around. That's why it's kind of nice to follow the data as we go forward with everybody, with all clinicians. Think of the data that we could actually, see these commonalities and correlations with together.
Thank you. Yes. And what kind of tubes will be needed, and how stable is the blood within the tube? Does it need to reach AONM'S within 2 days like ELISpots need to, or does one have a little bit longer?
So I think Marcus might be able to. Yeah, so the stability is like within 7 days is important.
Yeah.
That is excellent. So, if there were a little bit more of a delay, it wouldn't matter. The results would still be accurate. Marcus, anything to add to that? Perhaps you could tell us, Marcus, about the kind of tube that's needed. Well,
it's a serum tube. Okay. We use most of the times, and yeah, as Leona already mentioned, 7 days of transport is, well, it's possible from all around the world. Also, from when you send from Australia, usually we receive the blood faster than 7 days. So I don't see any issue in receiving the blood in time.
Oh, super. Okay. Well, there are, of course, a lot of requests for the slides and for the recording. These slides will be put up together with the recording on our webpage under past webinars, as always, within the next couple of days, and will also be sent out as a link to anybody who registered for the test originally. So it will all be made available. I'm not sure whether we'd like to say that, over Easter, there could be a little bit of a hiatus in dispatch. Obviously, next week we have, is AONM'S going to be closed for a couple of days, Marcus? Or if somebody sends in these bloods, for example, next Thursday, will you still be able to process them reliably, despite the Friday, Good Friday of Easter?
Well, we're still able to process the samples, as on Saturday is a regular working day in our lab. So it's not about the people working in the lab, because they will also be there on public holidays, but it's about the logistics, as we don't receive samples on public holidays. So
that makes
Friday
sense.
and Monday, we won't receive any samples from any logistic company, but even though we are working, but yeah. So Saturday is still a normal working day for everyone, for logistic company, and as well for us. So still possible if you want to send over Eastern. Yeah.
Yes, because of the 7 days actually that we have, which is fantastic for this particular test. Well, we will be making a note of all the questions under Q&A and chat that came in, for any that haven't yet been answered. We will also make sure that we have a sort of round table in the future, probably on a regular basis, of experts, though we'd love all of you to be there as well in order to help us with solutions. Though, of course, there are many that are well established, and, you know, even books out there on how to treat mycotoxins. But nevertheless, it would be very, very good to sort of synthesize that. So just to mention how very, very grateful we are to all of you for having taken the time today and for having developed this superb test. Thank you. And Marcus, it's very exciting to hear from you that you're going to be you know, collating the results together with the team. And of course, we'll support you in that. And also that you're developing a checklist. Tremendous. Dr. Schwarzbach couldn't attend today, but I'm sure he'll be there the next time. So thank you very, very much again for the time you've taken. And thank you everybody for having been there and taken an hour out of your time. Just to mention that on the, I think, 3rd of May, that's the first Wednesday in May, we will have Dr. Craig Shimazaki, the CEO of the laboratory in Oklahoma called Moleculera, that does the PANDAS testing for us of the antibodies to the basal ganglia for autoimmune neuropsychiatric conditions. And the title of that will be Infections and Our Immune Response. And that will particularly be picking up on the impact of Streptococcus A and scarlet fever, which have been circulating here in the UK, but also elsewhere, particularly during the winter, and have led to an increase in these very unfortunate, particularly PANDAS, that's pediatric autoimmune neuropsychiatric disorder, in these kinds of conditions, not just in children, but in adults too. So he'll be talking about that at 7 o'clock on that Wednesday, but of course we'll be sending out a notification. That was postponed from a few weeks ago, where it had to be delayed due to reasons beyond our control. So thank you very, very much again, everyone. Do send in your questions if you have any to info@aonm.org, and our team here will answer them. So thanks a lot again, everyone.
Thank you.
Bye-bye. Thank you.
Have a great evening. Bye.
Thank you. You too.
Thank you.
Bye.